There is some animal studies, though, that are consistent with what we showed. And this is using a mouse model, a swimming model. So they had mice treading water for 60 minutes, and it's a model of glycogen breakdown and fatigue. And then they extracted muscle out of the mice and exposed them ex vivo to glucose and insulin and different levels of ketones. So I think they had one with no ketones and then 1, 2, and 4 millimolar of ketones and measured glycogen levels. And this just demonstrating the model. So the 60-minute swim model did decrease glycogen, as you can see in the panel A. Panel B shows with no ketones, you're not getting any resynthesis. Or I guess that's 1 millimolar ketone, excuse me, no real difference. But with 2 and 4 millimolar, you're getting a progressive increase in glycogen synthesis over a 2-hour incubation period. So evidence, again, that ketones might have a role in restoring glycogen from exercise. And this has to do with the question of, can skeletal muscle produce ketones? So traditionally, we think ketone production or synthesis occurs in the liver, maybe to a smaller extent in the kidneys, but generally not to an appreciable amount in other tissues. So if you look at some of the enzymes in ketogenesis, they're very low in skeletal muscle, so Scott being one of them. And that kind of makes sense because skeletal muscle has a lot of different ways to utilize fatty acids. There's a range of different transporters to get fatty acids into skeletal muscle, so there's not a real need to use ketones like there is for the brain. So it makes sense the body would prioritize ketones for the brain and other tissues maybe, but not skeletal muscle. But very few people have done human work in a keto-adapted state and measured any kind of muscle adaptations. So this was one study we did that involved taking muscle biopsies out of individuals that were fed a ketogenic diet for 12 weeks and trained as well during that time period. And we had a control group that consumed a traditional high-carb diet. And we did demonstrate, my doctoral student at the time, Vin Miller, isolated mitochondria from those skeletal muscle and showed increased mitochondrial protein content and an enhanced ability of the mitochondria to utilize fat in particular. But interestingly, he also looked at ketone oxidation in those mitochondrial isolates and showed very, very low levels. So that's kind of consistent with this idea that muscle's probably not producing or utilizing ketones to any great extent. Not that it's zero, but a pretty low extent.